Plants don't absorb nutrients, they eat them. They actually eat bacteria. Fascinating, right? They eat bacteria through this process called rhizophagy, or rhizophagy, as some people would say. Professor James White at Rutgers University has been studying endophytes for 47 years, for as long as he's been married. He started looking at them as an undergraduate. I'm really excited to share this conversation with you about rhizophagy. We really simplify the process. We take a deep dive into what it is and why it's important, how it works, how you can support the rhizophagy process and interaction of plants. And we go a little bit even deeper into talking about what bacteria is and talking about what fungi is and the role they play and how it all kind of fits together. Dr. James White is an amazing human. He is easy to listen to, and this conversation is awesome, and I'm really excited to share it with you. I'm Natalie Forstbauer, founder and editor-in-chief of Heart and Soil magazine. If you haven't subscribed yet, you can head over to heartandsoilmagazine.com and get yourself a free issue just by signing up for the newsletter. Or you can subscribe for just $39.99 a year, where we amplify regenerative farming, gardening, and living for your health and planetary health. You make yourself an amazing day. Be sure to subscribe to this channel and like the video, share it with your friends to help it get seen and amplify the possibility of global regeneration and planetary health. Big love. Thank you for joining me.
You're welcome. It's my pleasure. I'm happy to.
We're talking endophytes and rhizophagy today, and I'm really excited about it because it sounds like it might be the way plants actually eat and feed themselves. And so I'm wondering, before we dive into what endophytes are and rhizophagy is, can you tell us what endophytes are and what role they play?
Yes. An endophyte is really a very general term. And all that it means is endo for internal, phyte means plant, internal plant. And it refers really to any kind of usually they're microorganisms. They could be bacteria and fungi that go into fungal tissues. But the term endophyte actually can be more extensive. For example, you have it can actually refer to plants like mistletoes and related to that go, a plant that actually goes inside another plant, so basically it's any organism that goes inside another plant. That's an endophyte. Wow. That's the term endophyte.
That's really cool. And so what role do endophytes play then if they are inside of a plant?
So plants use them.
Okay.
Plants use them to do things that they need. For example, there are some endophytes that will produce chemicals that will make the plant become more resistant to insect feeding, certain types of fungal endophytes. Other bacterial endophytes will go into plants, and the plant will interact with them to get these microbes to secrete nitrogen, because the microbe can fix nitrogen from the air, but the plant can't get it. So that if the plant has an endophyte that can fix nitrogen, a microbe, a bacterium that can fix nitrogen, then the plant, but indirectly, can get nitrogen from the air. So and all plants have these kind of endophytes in them.
That's really cool. So what I'm hearing is there can be nitrogen out here, and if the plant can't access it, then the endophyte can take that nitrogen and be a carrier and go into the plant, and then the plant goes, oh, there's nitrogen with that endophyte, and then it.
Yeah, well, so this is okay. So the rhizophagy cycle actually has microbes that are soil microbes that plants take into the roots, and then they extract nutrients from them, nitrogen plus metals, a lot of divalent metals, micronutrients, plus other macronutrients like phosphorus and potassium. So the endophytes, and which is extension of what we were talking about with nitrogen fixation, the endophytes can also carry other nutrients from the soil into the plant.
Wow. And so how do they do that exactly? Are they creating that themselves before they go into the plant, or are they picking that up as they go and then bringing it to the plant? How does that happen and work?
Both, both.
Okay.
They will pick some up, like these metals, like for example, a bacterium in the soil. They'll get these metals from the soil that just stick all over the wall, the cell walls of the bacterium, and then and also they will pick up these other kinds of nutrients, potassium and phosphorus and calcium and nitrogen and so forth. They'll pick those up, or either from other in the soil nutrients, or from other microbes. They'll get them, and then they'll carry those into the plant, or I should say, the plant will absorb these. They will attract these other bacteria to its roots, and then it will absorb them into the root cells, and then the plant will secrete reactive oxygen, superoxide, essentially superoxide, which is a very highly potent form of reactive oxygen. So the plant secretes superoxide onto these bacteria that it has absorbed into the roots, actually into the root cells. And then that exposure to superoxide will essentially oxidize off the cell walls, off of the microbes, off of the bacteria, and then the plant cells can absorb those nutrients that were on the cell walls. And also the bacteria will, some of them will be fully degraded, and so the plant then can absorb some of those nutrients from the bacteria that are fully degraded.
Mm-hmm.
Yeah.
That's really cool. So it's not like, because I think the way we used to think about plants and growing, and correct me if I'm wrong, the way I think we used to think about it, and I wish I had a fresh root here. I have an old root here. So
Okay.
the way we used to think about it is we used to think that the root of the plant would just absorb things, and they would go into, like, absorb nitrogen and
Right.
absorb
Right.
phosphorus and all the things that it needs, and then the plant would use it and it would do what it does. It would grow if it was getting everything that it needed. And so what I'm hearing you say is something a little bit different than that. Is that right?
It's very different, really. It's a piece that we didn't know was happening. And what we thought before, as you said, Natalie, is that plants, basically they're immobilized in the soil, and they simply absorb dissolved nutrients from the soil, and it goes into the roots, and then it spreads throughout the plant. But that's an incomplete story. It's not that that's wrong. That is correct, but it's not 100% correct. It's partially correct. What plants do is they actually have a much more active mechanism, or way, much more active. They're much more involved in getting those nutrients. So it's not just passively absorbing nutrients in the water. Instead, what the plants actually do, and this is extremely important because this is how plants get a lot of these nutrients that are hard to get. What they do is they cultivate bacteria around the roots by putting sugars out there. Something exudates, plants will make exudates and push them out of the roots. Those exudates are actually to feed the microbes and to attract the microbes to the roots. So they put these exudates out, the sugars. Sugars are cheap, by the way, for a plant because it does photosynthesis. So it's and that's from the sunlight. It can make energy and sugars, and it pushes those out. They're cheap for the plant, and then the microbes are attracted to that, and they come to the root, and then the plant actually will absorb those microbes, those bacteria, into the root cells, right at the root tip, really, and then it will hit them with superoxide to extract the nutrients. So it's an active process. So plants are doing this, and they can actually, depending on what they need, they can change what their exudates are, change the composition of the exudates to, for example, if they need nitrogen fixing microbes, because there's not enough nitrogen, what they will do is they will make sure their exudates will not have any nitrogen in it, or will have very limited amounts of nitrogen. And that will force the microbes around the roots to start fixing nitrogen. They'll cultivate the ones specifically that are able to get nitrogen from the air, and then they'll take those in. So this is plants actually are in control of the rhizophagy cycle. They're just not sitting there waiting for us to squirt fertilizer on them. This is what wild plants do. This is how they work. This is the actual functioning of the plants in nature, right? Our fields, if we do hydroponic or something like that, we expect, or in our mindset for agriculture is we have to fertilize the plants. But when you consider plants in the wild, they don't need any of that. They don't need any of our fertilizers. We mess them up when we put them on because they have these systems already going in them. They do rhizophagy, and then they have other endophytes that are fixing nitrogen in their tissues.
So we're discovering a whole new level of intelligence that plants
It is. Plants are much more. Plants are, I have it's exactly right. You're not the only one to talk about plant intelligence here. It comes up. A friend of mine owns a regenerative agriculture company, plant management company, actually said that almost the same thing, and that is that she didn't realize how smart plants were, and that is exactly what you're conveying here. That these plants are showing a type of intelligence, of plant intelligence that we didn't realize they had.
Well, it's actually a profound intelligence because it's so far beyond what our scope of understanding was, James, in terms of how plants grow and what they need to grow and to be healthy and to be strong and to be productive, to actually giving agency to a plant having the innate knowing of what it needs and saying, oh, like this little plant, this little root here saying, oh, I need nitrogen, so I'm not going to give you as much exudates, which is, I guess you could say, stuff that the plant lets go of into the soil, so that the bacteria around it can go, oh, I'm going to make some more nitrogen. I'm going to fixate nitrogen. And so that, and then the plant is like, oh, thanks for doing your job. And I'm going to take you back in and break you down and use that. That's mind blowing when you think about it.
It really is. And the reason, when we first discovered this process, when we worked it out, rhizophagy or rhizophagy, people pronounce it differently. We refer to it as a cycle, because actually the microbes, the plant is internalizing the microbes, and it's extracting nutrients, and then it's putting out survivors, and even cloning those surviving bacteria inside, putting them out to go get more nutrients out of the root hair tips. So it's kind of a fantastic process when you think about it. It's this rhizophagy cycle where the microbes are attracted to the root, they internalize them, they extract nutrients, and then they eject them back out at the root hair tips. And that is wow. You mean root hairs are ejecting microbes out, processing microbes internally and ejecting them back out to the soil. We always thought that the root hair, the only function of a root hair was to extend the surface area of the root so you could absorb more dissolved nutrients, but that's not correct. This is that's based on a misunderstanding of how plants actually work.
That's really powerful. And I love the way you named it as a misunderstanding, because it's not right or wrong. It's just where understanding is today, right? And so we are always learning. And I love that. So we used to think that these little root hairs right here, hopefully you can see it, these little root hairs, we used to think that they would grow out from the root to absorb more nutrients. And what we're learning is these little root hairs actually are letting go and putting that bacteria back into the
Natalie, that's right. if that what you touched was a root hair, that actually is a branch root or a
That's a
main root,
branch
and
root.
the root hairs would be on that. You could see if you were to
Oh, the
look at
even
that,
tinier ones.
they're tinier ones. Those are the
Oh, see
ones,
that
the
little-
little. Can you see them? I can't see them here, but they
Yeah,
would
can
be
you see
little
that if I hold it
tiny.
right there?
Yeah, I can't. A little bit. Yeah, I can see a little bit up there. I can see them up a little bit. Yeah, I can see them. Yeah, yeah, I do see. Those are the root hairs. Yeah, those are the root hairs there. I can
Oh,
barely see
amazing.
them.
Yeah,
I
they're
can barely
tiny,
see
right? They're
them. They're
tiny,
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 tiny.
tiny. They're tiny,
Oh
tiny,
man,
tiny. Yeah.
that's
Yeah.
really fascinating. So before we talk more about that cycle, tell me what that moment was like when you.
That was it was my jaw fell. Just it fell. We were at a microscope. One of my the lady, a young lady, but she's Catherine Kingsley. She's my laboratory main laboratory person. She manages my lab. We were sitting at a microscope and we were looking at these roots, trying to figure out, what the heck is going on, because we could see the reactive oxygen around the microbes in, but then we started noticing they were coming out of the root hair tips. And when we saw that, then that's when my jaw fell. It's a cycle. They go in at the root tip and they go out at the root hair tip. And it just, oh my gosh. Oh my gosh. And that's when I started to, we started really to look at it and try to figure out, what nutrients are involved and the details, exactly what's happening inside the plant and what forms of reactive oxygen were important. It really took us years to develop the techniques and the kind of histochemical stains that it was necessary to actually look at the chemicals that were being produced around these microbes to sort out this process. But it was astonishing. And since that time, I said, this is something really, to myself, this is something really, really important. And I promised myself that I would talk about this process to whoever wanted to know about it, to try to spread the word, because I thought this is so important that plants are doing this. It's necessary to get the word out. And normal science is very glacial, right? It goes very slowly. You write an article, you publish it, and maybe no one sees that article. And maybe, 30 years down the road, someone might discover it, and then they bring it out, and then they start, working on it, and someone else might discover it. You see, it's very slow. But this process is so radical, and I think so important for plants and how we cultivate plants, and agriculture, and developing clean agriculture using the soil biology and the plant biology to cultivate plants, to clean the environment, right? To make us have a cleaner agriculture. This was so important that I thought, I need to talk about, I need to tell everybody about this. And so that's what I've been doing. So I always agree to do a presentation if anyone wants it. And, so basically I've been doing that for years now.
That's awesome. And James, why is it so important to have a deeper understanding of rhizophagy?
Well, because right now we have the concept that most of us, have the idea that the way you grow plants is you put them in the soil, you put fungicides and insecticides around, and you put fertilizer on them, right? And the problems we have is that those chemicals are bad for the environment. Okay. About everything, almost everything there is bad for the environment. What the rhizophagy cycle and the role of endophytes tell us is that, in fact, plants, that's not how plants work. In fact, plants are much more active in using microbes in the soil to get the nutrients they need. And so we can, if we work on it, And people are already trying to do this in organic agriculture and in organic farming and in regenerative agriculture. They're already trying to use this. They call it soil biology, right? Try to use the soil biology to grow plants rather than all the chemistry. And the fact that plants are actually getting much of their nutrients, at least a key part of their nutrients, by oxidizing microbes in their roots and in other parts of their tissues, in other parts of the plant. Because they're doing that, that opens this other avenue to say we can now explore and develop the biology approach to growing our crops. And reduce all the contamination in the environment and the problems with human health and so forth.
Wow. So you're saying it's having a really big impact on the way that we grow food, and
it can have a big impact.
If we have a deeper understanding of it, do you think, or like what's different?
If we start to use it, if we use just the fact that plants do this opens the door to people exploring ways to, say, for example, fertilize plants using only the microbes, right? How do you then, for example, let's say you have a soil that doesn't have a lot of microbes in it. You can build up the soil to get a better, put more organic material in it, grow it, get a better soil, and then plants will have more microbes that they can then take in and get more nutrients. Okay, so that's kind of a, soil, building your soil, organic material, and your microbial diversity in the soil. That's important. The other thing is, okay, so you could fertilize plants with microbes. We know also that many of these microbes will interact with fungi, and they will colonize fungi. Many of the bacteria will colonize the fungi, and they will change the behavior of the fungi. They will, for example, if we remove all microbes from plants, and we don't have good bacterial concentrations in the soil, then you have fungi in the soil or on the plant, things like fusarium that will cause disease and kill the plants. But when the microbes are there, they colonize the hyphae of the fungus, and they alter the behavior of the fungus. They make the fungus less destructive, less pathogenic. In fact, in some cases with the bacteria there, fungi will become an endophyte, totally non-harmful endophyte, and grow in the plant and grow around the plant and benefit the plant in acquisition of nutrients. So we can control diseases with bacteria, with endophytes, and we can also, in some cases, control competitor plants, because some of the bacteria will go into competitors, weeds, for example, and reduce their growth. So you can have this, where the microbe promotes the growth of its host species, its host plant, crop plant, whatever it is, right? And then it will go into the soil, colonize as an endophyte this other competitor plant, but reduce its growth. And that is, that has to do with just an adaptation, right, on the host plant and not on these other plants. So, so you have these uses in cultivation. It really could. In fact, we think that it's possible. It may be possible to cultivate crops using only microbes, right, rather than other chemical applications. But there's also other things that one can do. For example, if you want to increase the soil microbiology, besides organic material, you could, for example, put something else, some other organic in the soil, and then that would increase the growth of the microbes, and then those would then be available to move into the plant. So you could essentially fertilize the plant that way, just by manipulating the microbiology. So that's the door we have.
That's really cool. So when you say using organic, like building the soil with organic material and microbes, what types of material would that be that we're putting into the soil? So how do we get organic material into the soil, and how do we how would we put microbes into the soil?
Well, the best way is to use a cover crop. And with the cover crop, you get a whole bunch of different cover crops, but you essentially you keep living roots in soil, and that cultivates the microbes. When the crop dies, that will be incorporated in to increase the organic material into the soil. So you do that, for a few years, and you build up that organic material, and at the same time you're building up the microbial diversity. There are ways to tweak it, and I mean, essentially, what do we do in agriculture with, we are normally just squirting nutrients out there, right, and put it in. Okay, that's a way to push a plant, but you're not using the microbiological aspect. You could do a similar pushing of a plant by, for example, incorporating some nutrients, and some people will do a little bit of molasses, or they'll do algal extracts, with a little bit of sugar mixed in, and they'll put that into the soils, and that'll encourage the microbes to, like a bloom of microbes. You'll get a short-term increase in microbial populations, and then those microbes will be there that the plant can then take to then get to for the nutrients. So it's possible to push plants using the microbiology. If you're thinking about if you start thinking about this mechanism, where the plants are absorbing the microbes to get nutrients. So you could actually manage a crop and plant growth by then managing the microbiology. So there's creative ways. There's a lot of people who are out there trying to think of ways to do this.
Yeah. So I have a question about pardon me?
Yeah. So this is the experimentation that people are doing, and if they know that, okay, the plant is absorbing the microbes, then it gives a confirmation that at this kind of microbial stimulation approach for stimulating, pushing crop growth, for facilitating or supporting crop growth.
Right. And so what I'm hearing from you is it brings the conversation of cover crops to a whole different place when we think about what types of cover crops we're using, if we're going to use legumes versus rye versus vetch. Would that be true?
It is true. And there are experts on cover crops, but all of these kind of cover crops will still stimulate microbes in the soil, because they're all going to be, essentially cultivating and absorbing microbes. So yeah, it does make a difference. There are experts on cover crops, and so if you go for a cover crop, you could talk to one of those people about which one would be optimal for your conditions. But the important thing is you have a cover crop. That's an important thing. Yeah, because that helps a lot.
Yeah, and that I thanks for presencing that, because a cover crop is the first place to start, and I'm just curious about where this can go and what we can learn from, because we have a certain understanding about the benefits of different types of cover crops, and imagine where the understanding could go even further with this understanding of the rhizophagy. What is the most important aspect of rhizophagy to have a deeper understanding of?
Oh, that's a good question. The most important aspects, I will tell you. Yeah. I mean, I don't know if this is the right answer to your particular question, but what is important to understand is that the plants need those microbes, those soil microbes. That's a key element. And then the other aspect is that the plants are really in charge of the rhizophagy cycle. That is, the plant is in control of that whole process. This is not a case where, it's like a pathogen, gets a plant and gets into a plant, gets into a wound or whatever it is that causes disease. It's not an arbitrary process. This is actually a very active process that plants are doing. And they're manipulating these microbes and extracting nutrients from them. And they're also putting them back out into the soil in higher numbers. So the plant is fortifying its rhizosphere, or soil around the roots, with these good microbes that it uses. Okay, those are 3 points. I think it's 3. But also what happens, because plants, when these bacteria are absorbed into root cells, plants are interacting with the microbes using reactive oxygen, this interaction to get the nutrients to come out. But what that makes the plant do, that makes the plant upregulate its own antioxidants and traits that make it resistant to its own reactive oxygen, its own reactive oxygen, its own superoxide and the hydrogen peroxide and other forms of reactive oxygen that develop because of that. But the plant becomes resistant to that. And what that means is the plant is also resistant to stress, because all stress is oxidative stress. It all converts into reactive oxygen in some way. And if you get this reactive oxygen all over the plant, then plants become stressed out. But if they're doing rhizophagy cycle, you're going to have more resistance to that stress. That it could, and stress could be, for example, high salt in the soil. It could be heat stress, wounding stress. If it's a yard or something that someone is doing a lot of I don't know, damage to this turf or soil, or the turf, right? The plants there, that any other kind of stress. Plants doing rhizophagy are more resistant than plants that you just feed with fertilizer. Those plants that are just fed with fertilizer are not going to show the same level of hardiness as plants with doing rhizophagy to get the nutrients. And you can also mess this up. If you put fertilizers on plants, what it will do is it will shut down the microbes that are fixing nitrogen. It will shut down that nitrogen fixing process. And so if that happens, plants may lose the microbes because well, I won't go into details there, but essentially you're truncating the normal system that plants use to get the nutrients from these endophytic bacteria. And then the other thing is, with more rhizophagy cycle, you're going to have more disease, less disease. And it's because those same microbes that are involved in rhizophagy cycle change the behavior of pathogens. Normally, pathogenic fungi, we don't think of fungi as having behavior, but they do. Anything they do, right? One behavior can be bad, we could call it virulence, and one could be good, we could call it symbiosis or non-virulence, right? We could call it that, non-virulence. And so these bacteria will actually push the fungus to non-virulence, to non-pathogenicity. And we've done many experiments where when you remove the microbes from the seeds and seedlings, they become highly vulnerable to fungal diseases. Their resistance basically breaks down completely without those bacteria. The bacteria are actually part of the immune system of the plant. Just like we know that our gut microbes have big effects on us, and we have to have a good mixed community there so that we don't have gut disease and other kinds of diseases, right? So the same thing with plants. They need their microbes, their endophytes, in order to give them that protection.
So what I'm hearing is there is a huge cost. The cost of using chemical fertilizers and
there is
inputs.
Yeah, there is.
A cost.
It's a big cost. It's a big cost, and we pay it, because we have to, when we pay that cost, right? I mean, that's the bottom line. We have to eat. So, but all that's based on really the idea that it's all about the chemistry, and that really is not correct. Yes, it's not. It's only partially correct. Right. It's only partially correct. Yes, we can manage plants with chemistry, but there are some other problems. Then we have diseases that become more significant. We have more fertilizer we have to put on. Diseases are there. Then we've got to put more fungicides there. And plants are not as well. I mean, it goes right down to herbicides. Even herbicides. Herbicides become a problem too.
That's interesting.
Yeah.
That's interesting. The image that's coming to me is plants arrive, the seedling arrives, or the seed awakes, and it has this massive amount of innate intelligence. And given the right environment and the microbes and all the things, it actually has the ability to grow and flourish. And every single time we put in something like a chemical or fertilizer, then we're actually taking away the agency. So it's almost like not just that a plant is getting artificial intelligence and artificial support to grow. We're actually shutting down its intelligence to know what to do to provide for itself. It's like teaching somebody how to fish rather than giving them a fish, right?
What we do, I like to put it a different way. I put it, what we do is we in a sense invalidate the systems that plants are naturally using to get nutrients. When we put chemicals on, they can get those nutrients, there's no doubt about it. But what we do to the plant is we take away its capability to get nutrients from the bacteria how they normally get microbes and nutrients, use by oxidizing these bacteria. Now, some of these bacteria, I should say, there's two sources for those microbes, and one source is in the seed. And we know that some of these bacteria vector on and within the seed itself. And other bacteria are absorbed directly out of the soil. So you have multiple sources for microbes, right? And plants are taking in both sources to get the ones they go through the seed, some will stay in the plant, some will then go into roots and go out into the soil and increase in the soil, so that when a seed is formed, ideally, if it's in a healthy plant that has all its microbes, you'll have bacteria inside the seed and on the surface of the seed. They'll be on tissues that stick on the seed. They'll be there. And so wherever that seed goes, it is already invested with the microbes that it will need to get a start, right? To start growing there. Of course, it can then attract other microbes that go into it, but they really have these one set of microbes that they carry with them. That's all.
That's super powerful.
Yeah.
So the seeds are literally formed with the microbes that they need to get a start. So what does that so one of the things that we do a lot, or some people do more often in organic farming and biodynamic farming, is sometimes we'll do seed soaks, and Korean natural farming does seed soaks as well in different ways. And so is that something that helps or inhibits the seed, or do you know?
It depends on what you soak it in, right? If you soak it in Clorox or something that would kill microbes, this is a very bad thing to do.
So that is actually a practice, because I'd never heard about that before. And I was listening to a podcast the other day, and they're talking about sterilizing seeds. And maybe I'm sheltered because I grew up more in an organic farming environment, so I'd never come across that before. But that is actually a practice.
Yeah, that's a bad practice.
Wow.
It's a bad practice. It's a bad practice. It's a bad practice. Yeah. If you put your seeds then in a very clean environment, in a very organic, microbial rich environment, they can recapture those microbes, or at least some of the microbes. But right away, they may not be able to get exact ones. Right away, you've put the seedling at a disadvantage. Now, I mean, people may have, well, I mean, here's another issue with many of our crops. Many of our crops, the way that we've domesticated plants has tended to cause the loss of part of the microbiome on seeds. I mean, I'll give you an example. what happens normally with seeds is that the plant will oftentimes, with many seeds, will either hold those seeds in the environment where they're going to get some moisture. I mean, consider something like corn. You get a corn cob there, the wild corn, right? It would stay on the cob, and it would stay in the weather, and it would stay there for a long, for months before they fall to the ground and then start to grow in the next spring, right?
Mhm.
But what people do, and we do this with all our seeds, and it causes a problem, but it seems natural to do if you're not thinking about the microbiome of the seed. You gather them soon as they're ripe, the seeds, and then you put them in a cool, dry place. And what that does is that prevents the microbiome of the seed from fully developing. So it doesn't develop, right? So
Mm.
it would need this moisture, moisture conditions. It would need maybe to fall to the ground and to lay in the dirt, on the earth, on the soil, and collecting microbes. So you see the microbiome takes a development, or takes ripening. The microbes, if there's enough moisture around, the microbes can divide, and the tissues that the plant has on the seed for them, they could divide, and then they're ready to, they have full microbiome, they're ready to grow, and you won't have disease. But because we've removed part of the microbes, that makes plant seeds more vulnerable. So you have things like fungi then that can colonize a seedling and knock it out real easy, because the microbes aren't there. The bacteria in specific, antifungal, right? They're not really there.
Mhm.
We of course use chemicals to control that, all
Right.
of that, for growth stimulation and everything for corn. But the indigenous Americans, they were the ones um, who actually, in Mexico and the areas, who actually domesticated corn. And
Mhm.
the indigenous Americans had their own way of using biology to grow the
Mhm.
plant. And what they would do actually is they would, like I said, they would store their seed, they would collect their seed, and they would store their seed in a place that was dry and cool.
Mhm.
If it at all possible to find a dry, cool place. Sometimes it wasn't possible, but typically they would. And then they would take the seed, and they would then inoculate it with bacteria from wild plants. And the way that they would do that is they would go out into the woodlands, North America. This is called Iroquois corn medicine, is what they called it. Iroquois corn medicine. But they would go out and get these wild grasses, things like Elymus, or Hystrix, the common grasses in the area. These are and Phragmites too. This is another genus of grass, but these are all wild grasses that are common in North America. And they would take the roots, and they put it in water, and they would beat up the roots, get the microbes off of them, and they would even heat that a little bit, warm it up a little bit. And then they would take their seeds, or the corn seeds, the corn kernels, and they would put it in the water, a little bit of water, and they would let it germinate a little bit till the root came out and got colonized, because they
Mhm.
didn't know there was any microbes there at all. They didn't know anything
Mhm.
about that. They thought this was magic. Okay,
Mhm.
this was magic for them, Iroquois corn medicine. But then they would take the corn that partially germinated, and they would plant it in their rows, in their field, or in their mounds, however they were doing it. They were doing three sister agriculture, whatever they were doing. They would put them there, and then that would recover some of the microbes that had been lost, in the way that we're handling our seeds. And so, to go back to the problem of people sterilizing, the reason they're sterilizing is because they lost a lot of the bacteria on those seeds that would actually that would control the fungi that they have there. So they're left trying to kill the fungi. This is kind of
Hmm.
it seems counterintuitive, right? It seems counterintuitive. One microbe is a problem, so you put other microbes on. But that's what needs to happen, is the seed needs to have more more microbes that would actually control the pathogen, rather than doing something like sterilizing seeds, which kills everything. It's a kind of, it's almost like if we took a bunch of antibiotic, right, and killed their gut microbiome. That's not a good thing to do.
Right. So what I'm hearing and understanding is the rhizophagy might happen and start under, like, in the soil. The beginning of the cycle, or maybe even just part of the cycle, is actually in the seed itself. So the seed needs to be in good integrity and have a healthy microbiome and have all the microbes heading into
not all of them, but there is a certain number that a certain community that's vectored inside the seed near the embryo, just around the embryo. We can see them in there in sections, like tomato. We've done sections of some of these, I think mints and tomatoes, and we could see the bacteria in there in the sections. Also, some of them you can't remove from sterilizing. That way that's how we know they're in there. But others are vectored right on the seed surface. And so you have tissues like some grasses that have little lemmas and tissues, little leaf tissues that stick on the seed, and those actually carry microbes on them.
That's so cool. So James, is a hot water bath, because some people soak their seeds in a hot water bath, which is different than Clorox or sterilizing, from what I understand. Is that something that will damage the microbiology of the seed, a warm water bath, or is, like, should we avoid that? Or
warm water bath is not going to hurt. No, it's not going to hurt. That's not going to hurt. These microbes, many of the microbes on the seeds are in a biofilm form, right? And they're pretty resistant. They'll last dry. They'll last. I mean, we've had seed of grasses like tall fescue that are still viable with their microbiome stored in the refrigerator for a long period of time. I think we're going on at least 10 years or 12 years for some of our seed supply. The microbiomes will last a good amount of time, so long as the seed is dormant. Now, there is an issue with the death of the microbiome. And for example, some of the decline that happens in seeds as the germination percentage goes down, people figure, well, the embryo's dying in there, and so the germination percent is going down. But it's been shown that you can put endophytic bacteria back on those seeds and recover the germination percent. So you bring them back up. In fact, a friend of mine at another university actually did a patent using one of the endophytes that he did as a means to rejuvenate old wheat seeds and other seeds. So
that's neat.
Yeah.
That's really cool. So if you were to describe rhizophagy or rhizophagy to a 5-year-old, how would you explain it to them?
Plants eating bacteria. That's what I would say. That's what I'd say. Rhizophagy, rhizo root, phagy means eating. Phagy means eating. Rhizo root, so it's root eating. And this is named by an Australian group, Queensland, Australia, who really identified the process. And then they gave it the name rhizophagy. And then we took it from there and started to understand the biology and how it worked. So this Australian group really gets the credit for first pointing that out in a paper in 2010. We were actually working on this around that time, working on this oxidative extraction of nutrients in roots and on roots, but they get the credit. And hence, when we understood it was a cycle, the only thing to do is then to take rhizophagy and tag the cycle on it, right? Put the cycle in the term, and then in order to explain what's going on in more detail. So it's
really
roots eating soil microbes. That's it. I mean, that's simply what it is. And they digest them using reactive oxygen, using superoxide, which is also cheap for the plant because it gets oxygen right out of the air. It uses the oxygen to get the superoxide, then it can zap these microbes with. It's really cool.
right. So does that mean so where does that leave a plant on the like vegan carnivore
That's a good
scale?
question. Well, I think they are, well, they're not eating animals unless you consider bacteria animals, or, they also will take in yeasts into their cells too. So yeasts can be part of the rhizophagy process. They can be absorbed in there. We can see them and we've used them. We've done lots of experiments. So they're eating fungi. I guess vegans can eat fungi. They're eating bacteria. I guess that would be like, I don't know, eating if we eat yogurt, can we be I guess we're not truly a
You're
vegan.
vegetarian, but not vegan.
Not a vegan. We could be a vegetarian, right? If we're eating yogurt.
Yeah.
All right. So if we're a vegetarian, so plants would they be vegetarians? Yeah, but it's also possible, and we don't know, and I've never seen it, but it could be possible that plants are absorbing more microbes than just bacteria and fungi, and that we don't know. We don't, in my lab, we don't work with the protozoans as much. But I mean, they could, who knows, I don't know what they could be absorbing. And if they start eating protozoans, then you're something right at the foot of the animal line, right? That goes to animals. And then you say, well, they're eating animals, right? They're eating animals. If they're doing that. We don't know if they're doing that.
Yeah, we don't know yet.
Yeah, then they would be meat eaters, carnivores. Then they'd be carnivores.
Oh, that's fascinating. So just to help pull things a little bit apart. So just again, I'm a 5-year-old. Can you please tell me what bacteria are, fungi are, micro, and just pull apart the big parts for me.
I will. Bacteria are some of the simplest organisms that exist. They are simple organization, and they're tiny. You don't get giant bacteria because they're kind of simple. So they're the simplest really among the simplest organisms, the bacteria. But there also may be other groups. I mean, when I say bacteria, I'm not parsing out different groups, but it could be more complicated if we did that. So they're the simplest organisms. They've always been on Earth. Before the plants came about, and the animals came about, we had bacteria. So more than likely, the early developing plants and animals they came from these microbes, right? They came from essentially bacteria, archaea, these simple organisms, right? They came, and they were using bacteria, and we know that because plants use bacteria now on their roots, and we could look at the very primitive ones like mosses. Some of the simplest plants, right? They have bacteria all in their tissues. So very likely, plants have always had them. Animals, it's the same deal. They evolved, they developed in a primordial soup, right? With all this stuff in them that they could feed on with bacteria and everything else. But when they left that primordial soup, we carried that primordial soup with us in our bodies, in our guts, right? That's the primordial soup, all those microbes in there. This is where plants and animals came from initially. So they had them there. Okay, but now fungi, that's another microbe. It's more complex than the bacteria, though. Typically they're the ones like your mushrooms, right? Mushrooms, but they have this smaller phase of hyphal mycelial phase which is just tubular growth in the soil or in the plant or in whatever it's degrading in wood that it's breaking down. That is the fungus, and that's fungal. So they're a completely different group, more advanced, but still small enough that plants can suck them in and get nutrients out of them. And so, so those two fungi, bacteria. What else did you say, Natalie? I can't remember what else.
The main players in the
yeah,
soil, like that's happening
yeah,
with that the plants are interacting with.
that's them.
Thank
Yeah.
you. And then to help people understand, like mycorrhiza fungi fall into the fungi. So then, and the
Yeah.
protozoa, like where
Yeah.
do they
I
fall in?
will tell you. It's a good question. So, so plants get their nutrients in multiple ways, right?
Mhm.
We know, we've always believed that plants were getting nutrients dissolved in soil water, just we've always known that. And that's why our fertilizers work, because it goes right into soil water and they can absorb them from the soil water.
Mhm.
We a few years ago, maybe a century ago or so, it became clear that plants were also connecting with fungi, and there are certain types of fungi called mycorrhizae that will go into the roots. This is what plants do. This is how another way that plants get nutrients is they'll hook up with mycorrhizal fungi, and then those hyphae will go out into the soil, and then the fungi then absorb nutrients, interact with bacteria in the soil, and get nutrients from them. And so there's this, essentially the mycorrhizae then will get those nutrients and bring them back to the plant, because that's another way plants are getting nutrients, right? The third way is that they're absorbing bacteria directly and oxidizing them. So that's the third main route of nutrient absorption in plants. That's the part that wasn't known at all.
Which is the rhizophagy, which we've
Rhizophagy cycle. That's the rhizophagy that we're talking about. Yeah, that's the new thing.
Awesome. And so knowing what we know now, knowing what we're discovering now, what are the next questions and what are the next conversations we might want to be having with ourselves and with our colleagues and with our friends and our family as we are deepening into understanding the intelligence of these plants that we
that's a good question. What I think is the next thing. I mean, we're obviously looking at details in my lab, trying to figure out, how everything works, right? And trying to measure precisely what nutrients are being oxidized from these microbes, right? And it turns out every microbe gives different nutrients. There's not the same one for any microbe. So they have different capabilities in terms of, so plants are getting different ones, right? I mean, that's the case. There's a lot of details that have to be figured out. And a lot of this is on a science lab level, right? That most of us who don't have labs and wouldn't be able to do. There's a lot of that, but there are aspects that are not on that level that really require people to experiment and figure out how they can use they can manipulate the microbiome, manage the microbiome of the soil and the plant in order to cultivate crops. And doing techniques like, for example, there are people who do ferments, and you get products, or some products, or people do ferments on their own, or they'll get microbes, they'll get compost washes and those things that are a bunch of microbes together, and they'll put those around the plant or on the plant. Maybe even on the leaves, right? Because that actually will stimulate the, microbial entry into plants. And we didn't talk about that, but the plants can get in on the surface of the leaves around the meristem and so forth, so they can get those microbes in. But you put those things onto the soil, putting, for example, different types of microbial, either nutrient solution to stimulate the microbes, or a solution, a suspension of microbes from any of those kind of ferments, or compost washes or whatever, to put the microbes there. So these, and also the ways that you treat the plant, for example, the seeds, to get those microbes functioning, ways to restore microbiomes. I mean, all of this, there's a lot that has to be done that scientists in their laboratories are not going to be able to do. This is going to really require a broad base of human exploration, individuals and companies. There are a lot of companies that are trying to do this. They're regenerative agriculture companies. So they're trying to find new ways to stimulate the microbiome and then thus stimulate microbial growth. So it is, but it's not something that I'm, for example, going to be able to do for us. The best thing that I can do is to, keep studying the details and to let everybody know what we're finding. But it's going to take all of us, to create a new agriculture, right? A new biological agriculture. It's going to take all of us. We don't know what the limitations are. Can we do it in all kinds of soils? There may be some soils, deserts, for example, if we're trying to bloom a desert, it may be hard to do this because there's nothing in the soil. It's almost, but you get my point. We don't know where the limits are. We don't know exactly what are the techniques that are going to be useful. We know some things that people have done that seem to be working. And the reason they seem to be working is because plants get nutrients from those microbes and other benefits from those microbes. But, it opens a door.
It sure does. It opens the door wide open. And I love that because, the gardener can do it at home just by doing compost washes, and it leads right into big ag that can be doing it with just changing the way they do things, even introducing cover crops or.
There's a book also, Natalie, that I don't know if you're familiar with Jeff Lowenfels.
I just interviewed Jeff this week.
Just interviewed Jeff. Did he talk about teaming with bacteria?
Yeah, that's why that's why I wanted to have you guys in the next magazine together. So
it'd be good. Jeff did a really good job on his book with the biology. It's reachable, right? He did. Your guests already, your people who are your audience already knows, because you did it last week, but he, the basic biology, he did a great job on that, explaining it. He writes very well. It's a book. It's not a thick book, so it's easily read. And teaming with bacteria is what it's called. And I think it's not expensive.
No, it's not. Jeff, I have such a deep appreciation for Jeff because his books are easy to read, they're easy to understand, and they can meet a kindergarten teacher all the way to a farmer who has been farming for decades or is fifth generation, and to a person who absolutely knows nothing. And so this is his book, Teaming with Microbes, which I have. And so this is the thickness of it. And it's just, like you said, it has pictures and it's simple and it's easy to understand. So thanks for presenting that.
You're welcome. That's the Teaming with Microbes, right? But
that's right. This is Teaming with Microbes.
And the one that's out is Teaming with Bacteria. That's coming out. It's already out in Australia. He said, I think he told me it was the 26th or something like that. It's supposed to be out, or 27th of this month.
Yeah. So Teaming with Bacteria. Brand new.
Bacteria, brand new.
And it has all your groundbreaking work in it. So that's really exciting.
It is.
What's most important to you about the work you're doing? Why is it so important?
Because I think it changes the way we think about plants and plant cultivation. I think it changes things. It opens, like I said, it opens a door for everybody. Now it takes people, it's hard for people to, it seems so radical. When you think about it, what we're saying is something that is really hard for a lot of people, and that is that plants are absorbing bacteria into their cells, right? That's a door that a lot of people is so new to a lot of people, they can't conceive of it. I've looked at, people before we described it, before the Australian group discussed it, before we described it, to the extent that we described it also in many different plants, there wasn't much known about, absorbing into the cells, actually into the root cells. That is, that is radically different. And so people just have not, there's like a barrier. It's like blocks on eyes, and I can look, for example, many papers, and you could see these things very easily in root hairs as they're ejected, and we can watch them being ejected from the root hairs. But in the photographs, in the literature, people will point to them inside the root cells, and they call them vesicles. And we could see those from the pictures. We could see the bacteria also accumulating at the tips where they were being ejected, and they never noticed them. They never called these bacteria. They only called them vesicles, and then they ignore what happens onto the root tip surface. It's because we're blind until you see it the first time. We're blind. This is like nature. We think we have everything figured out, but we really don't. In fact, we don't even see what we don't understand. We don't see it. We're blind to it. And that's the phenomenon. But you can look in the literature and you can see that they were there all along. People just didn't recognize them.
And when you think about the work that you're doing and how meaningful and important it is to you and to what we're discovering in the way that plants interact with their surroundings and how we interact with each other, what is the most exciting part of that and most meaningful part of that when you think about your friends and your family and the impact that has on the people you most deeply care about?
That's a deep question, Natalie. Can you rephrase that? Can you rephrase that in another way so that I can?
Yeah.
I can't quite.
Yeah. Well, I'm
comprehend that
curious. You clearly have a deep passion about and excitement. You have a deep excitement about the intelligence of plants and what we're learning about them. And I'm curious, when you think about that understanding and it going beyond the lab, beyond the microscope, into the ethos of your friends and your family and humanity and community and the earth, what is that about for you? What drives you?
It does not. I am the same person I was. I mean, a derivative of the same person I was when I started as a young professor, right? Or as a graduate student doing the same thing. This is merely an extension of my role in life, right? Is to, I've always worked on endophytes my entire career. I've done some fossil work, but they're still microbes in plants, right? From early evolution of life and on the planet. But this is merely an extension of what I've been doing all along. I can tell you that nobody notices. My family and friends, they don't. It's the same, right? Look, in science, it's ho-hum because it's just another discovery. But it so happens that I think it's so important that I keep talking about it because I do think that it's important for how we grow plants and for the sustainability of agriculture and so forth. But it is, I mean, this is a standard thing that, so, I do a lot more talking than most professors, but this is what we do. And so, as a university professor at Rutgers University, this is my job, right? This is my job. And everybody around me knows that. I'm the same person I was, when I was a young professor. It's the same deal. Just now that I just talk a little bit more and a little bit more excited about, I was excited then, oh my gosh, I've had a wonderful life doing this, so, and it is, just now, I think we've hit on something, an area that is really important for everybody to understand that it's happening, right? That it's happening.
Yeah. And why endophytes? Why did you decide to do endophytes at the very beginning?
Oh, it's the road less traveled, right? It's the road less traveled, right? Here's the thing. When I was an undergraduate at Auburn University in Alabama, right, I had one of my work study, because my father was military, so we didn't have a lot of money. One of my work study jobs was working with a scientist who was studying fungal endophyte in forage in tall fescue, because it was so important. Cattle, they do a lot of cattle out there. And so I started looking, I worked with him, and I started, screening grasses to find these endophytes using a microscope, just a undergraduate student, a kid there sitting in the lab with a microscope looking at all this stuff. And I came to realize that no one really is studying this. Nobody really is studying this. So when I went on to do my PhD at the University of Texas, I said, okay, I'm going to extend this knowledge. And I started to then look at a lot of different grasses for these fungal endophytes. And so that, that first paper that I did, those first papers that I did as a graduate student, had really hard to do that, right? I had to ended up going into debt to publish the article on it and all that, on our family credit card, and do field work and all that, but it paid off. It was an important article that paid off. And so this whole fungal endophyte thing picked up, and it was part of creating all the interest and excitement around fungal endophytes, right? And then about 12 years or more ago, I realized that fungal endophytes, they're there, but there are many more bacterial endophytes, and we have to understand those. So we started working on these bacterial endophytes to figure that out. All plants have bacterial endophytes. Only some plants will have fungal endophytes too. So we wanted to then look at the bacterial endophytes and understand them. So basically that's how this developed, and it just natural. But this is something that university professors do. That's why people keep us to do this, right?
That's
I
right.
mean, it's not.
We appreciate you.
Yeah. Well, thank you, thank you. This is what we do. This is what we're supposed to do. This is why you keep us, and everyone else keeps university professors. That's the reason why you keep have a university, because you have these people who will do these crazy things, and develop some understandings that we don't already have. Yeah.
Is there anything we didn't talk about today that was on your heart that was really important?
Well, I'm working with a plant breeder, Walter Goldstein, and he's trying to breed corn, actually, that it has nitrogen fixing endophytes in it. And so we are actually helping him to do that. And there's some really cool stuff about those, these land races, seeing the microbes in there and transferring them, so we're working to try to transfer them into corn, to try to develop lines of corn that will fix their own nitrogen. And how you have to develop, how you have to handle the seed, moving the microbes between the lines of corn to get them, because we've lost them in a lot of our cultivars, our hybrids, anything tissue cultured, you screw up the microbes right away, because that you usually sterilize tissue to do tissue culture, and those microbes get lost. So trying to replace those microbes back into our crops, and this is something I'm kind of excited about, that because that would be a direct way that we could impact agriculture. And not everybody can do it, because it takes some, we use microscopy, a lot of microscopy and histochemistry, and very few other people are doing that to see these microbes and to track the microbes. So I'm kind of excited about that. Maybe we can create some new cultivars that will be some corn. And corn is a big crop, important crop that people could grow with, wouldn't have to need any fertilizer on. That would be or at least no nitrogen fertilizer.
Yeah.
And, yeah. So
That would
that's,
be
I'm
amazing.
kind of
And,
excited about that.
and just to clarify, that's not genetically modified. That's
Oh,
just through.
no, that's moving
Yeah.
bacteria. That's moving
Yeah.
bacteria between the plants. So creating, I mean, I say creating, but we're recreating, you know, and standard breeding methods, and then moving the bacteria from the land races, you know, that are actually out of places like Mexico and Peru, right? Moving those microbes. And some of them you can't even cultivate, you know, they may be non-culturable. So developing the techniques to move them back into our corn. And then Walter would have to do the Goldstein at the Mandamin Institute, you know, where they're breeding this corn. They'd have to then make crosses to stabilize the microbiome and so forth, and then develop the ways to support that symbiosis, right? So that
Right.
it's really exciting.
Fascinating.
Yeah, yeah. And
It's a
nobody's doing it. They don't know how. They don't know how to do it. But we think we have our foot in the door, you know, little toe right in the door. So we're trying to do that.
Yeah, well, standing with you while you figure that out.
Yeah. Thank
Yeah.
you.
Yeah. Well, thank you so much, James. Thank you for your time and sharing just a small part of your story and what you've
Yes.
discovered, which is probably one of the most profound to date. And I can't wait to see what you unearth as you move forward.
You're so welcome, Natalie. It's my pleasure.
Yeah, thank you. Grateful you joined us for that conversation and interview. If you haven't subscribed to Heart and Soil magazine yet, head over to heartandsoilmagazine.com. Click on that subscribe button and join us for just $39.99 a year. You make yourself an amazing day, and I'm really grateful you're part of our community.